It's recording now. Okay. So what's a base pair? So this is the base, now we discussed. So base pairs, why do we say base pairs? Because in genes, there are two strands, okay? So in one strand, if A appears here, T would appear in the other side, okay? So this is one base, and this is the base pair, okay? But when we are writing the sequence, we only write bases in one strand only, okay? A, B, C, G. So accordingly, there's another missing information written here, but everybody knows for any nucleotide sequence like this, there's another sequence which is the complementary pair, okay? In the complementary strand. So only single strand nucleotide sequence only we are writing here, okay? So here like this. So these are about nucleic acids. So how about proteins? So I'll erase this and talk about the protein. So in proteins also we have there are 20 basic units. So if you have read about that, there are more than 20. So although we are considering 20 for this course. So for the 20 standard subunits or amino acids, they can form different proteins. So a protein sequence, we call a protein sequence when different amino acids makes a sequence like this X, U, A, whatever it is. Okay, so there are 20 different components, and they get the components of amino acid, they get together and create a sequence. So it's called the protein sequence. So this is a protein sequence. So that's about the sequences. There are mainly two types: protein sequences and amino acids. Nucleic acid sequence. Protein sequences are made up of nucleic acids. So you know that nucleic acids get together and create a protein, and create a gene, and amino acids get together and create a protein. So those are the basic things you need to know. So I will share my screen again for you. So you can see here DNA sequence data, which are actually nucleic acids. How many sequences with the year? So this is the year is not visible here. However, in the time increases, you can see I think you still can't see. So if you can't hear me, you have to let me know, okay? So that's how can I know? I mean, I can't teach to people who don't talk. Okay. In the first year, I'm doing with first years, they are very active. They talk and they whatever the thing required, they do it before I ask even. So there should be one person who is, always answering, because most of the time when many people talk, it's a disturbance. However, you can appoint one person to answer one day or something like that. Maybe today let's appoint who would like to talk and continuously talk. I mean, just to say yes or no. Do you hear me?
Yes.
Okay. Okay, I mean, there should, from the next day, you can appoint one person. Otherwise, I mean, it's so boring talking with the computer. I don't know.
Okay.
Okay. So, So that's why actually we need uh, help from the computer science side to store this amount of huge vast data. So you can see it's increasing rapidly, and recently also I looked at these websites, how these things evolve. This data doubles in every 18 months in GenBank. GenBank is a database where it stores genes. So it said for once 18 months, the data doubles. Okay, so that's why we need databases. So who keep this databases and how these databases are updated and how the data are analyzed and kept. So that's the next problem. So so this is about the public domain bioinformatics facilities available. So there are three main databases hosted by main three countries, developed three countries in the world: United States, United Kingdom, and Japan. So they are like the kings in the world. Okay. So they freely host these databases and these servers, and they keep this data and analyze this data using their money. So, so we can download these whatever the information you require from these databases. So let's discuss now about how the data are uploaded to these databases and how can we retrieve this valuable information. So, so these are the three main databases. So if you are an experimentalist, biologist who find out about these genes, and assume that now you have find out a new gene. Okay, now you need to upload this to a database for further analysis. So for that, you have three options. You can upload to one of these databases. So these are called primary databases, mainly because you are uploading primary sequences. So what are the primary data? Primary data is the sequence, whatever the nucleic acid or protein sequence, that's raw. Okay, you just take from the particular organism, and you have just set of letters representing nucleic acid sequence or a protein sequence. So then you upload this to either in CBI, EBI, or KEGG or DDBG. So these are the main three databases available. Okay. So, so these databases, so these are actually not only hold data, they can also analyze data, and also they provide analysis tools as well. So let's talk about analysis tools later. So they have databases and analysis tools. So these three people have databases and analysis tools along with these huge archives. So let's now talk about these archives. So these archives, these three archives, NCBI, EBI, and DDBG and KEGG. So these are actually these have massive biological experimental data. And these biological information databases can be classified into two types: first level databases or primary databases, which come from the raw data. Okay, so they are taken from the experiments directly and uploaded by the biologists. Okay, they are simple and private. And the second level databases, the derived and analyzed data actually from this primary data are called the second level data. And as an example, second data structures, hydrophobicity plots, and domain specified sequences are stored in the secondary databases. So to have a better understanding, so what is this hydrophobicity? So once assume that you are an experimentalist, okay, and you have now find out a new gene, and you are uploading that to a database, one of the three databases hosted in United Kingdom, USA, and Japan. Okay, so once you upload this data, the people can analyze that. Okay, it's publicly available, so people can analyze data. So there are different analysis methods. So one analysis is this, looking at the hydrophobicity. So what is hydrophobicity? So if you look at the web, and I think you have learned this as well, hydrophobicity is the likeliness to water. So some sequences, they would attach with water because they have some nucleic acids which like water. And some sequences, they repel water. They would not mix with water, mainly because they have some components which do not adhere with water. Okay. So these two types of sequences or proteins, protein sequences are identified. So you have done some analysis. Okay. So to the primary data, you have done some analysis, and now you have come into a decision that this particular protein is hydrophobic or not. Okay, so that data is stored as secondary level data. Okay, so they are primary level data. Primary data is actually something you have done experiment. You have done experiment with some organism and found a protein or a gene. So that's primary. Okay. Secondary level, you have to do some analysis on those particular data. So this, I think now you have some distinction between this first level data sets and second level data sets, databases or datasets. So, these three databases are primary databases. However, they host some other databases also, which should store these secondary level data. Okay, so if you go to NCBI website, you will see there are not only one database available, there are several databases available for you to upload data. Okay, so that's what the primary database and secondary databases are. Do you have any problems? Can you actually hear me? It's raining.
Yes, ma'am.
Yes, ma'am.
What did you say?
Yes, yes.
We can hear you, ma'am.
Okay. It's raining so hard, so that's why there are disturbances. However, we'll continue. So these biological database can be categorized. Okay. The databases which carry out information about genomes. So what's a genome? Can one tell me what a genome is? What's a genome? I think we discussed on the first day what a genome is.
It's like that, madam, a collection of whole gene information.
Yes, whole gene. Whatever the whole information of genes is called the genome. All the genes in a particular organism is there in the genome. Okay. So as an example, we can say COVID-19 genome. So let's look at the genome today. Okay. So that's a very important hot topic these days. So let's look at, let's try to download the genome of COVID-19 today. So that's one of our goals. So there are genome databases which carry the whole genome, the whole set of genes in a particular organism. Okay, so there are the different types. Ensemble is one, genome is one, UCSC is one. Okay, so there are three main genome databases available. There are more than this. However, this here it lists only the important databases and the protein databases which should carry out, which should store protein information. UniProt and Protein are two main popular databases where you can publicly available databases where you can freely download any information about proteins you wish. An expression database that would store information about the expression of proteins, genes. So where does the expression starts, where does it end, and which kind of protein would be expressed from a particular gene. So those information are stored in the expression databases. And the structured databases would store the structurally related data, like how a protein would fold and create a structure, what the protein structure would look like. So we'll be going into these databases and having a look at inside some important genes, proteins today, okay? So protein structure mainly is stored in this PDB and SCOP databases. And the interaction databases would include data about how these proteins interact each other. And the pathway databases would include information about the pathways. So what's the pathways? Pathway, let's look at about this one now. So what a pathway is actually, I use the whiteboard to discuss what a pathway is. I think you can see the whiteboard, isn't it?
Yes, madam.
Okay. So a pathway is actually, so if I draw a pathway. So assume that you are exposed to UV radiation, okay? So you know UV radiation can create, it's very harmful to you and it can create what? Skin cancer, okay? So in UV radiation actually, so if it incident on your skin, so what are in the skin? There are skin cells. In skin cells, so in each and every cell, so if we zoom it out, so in any cell, so if you look at the inner structure, there's a nucleus. So in the nucleus, what you have got is the whole lot of genes which we are composed of. So this UV can actually create mutations in these genes. Okay, so it can create, so these are the chromosome pairs. So it can create changes in the nucleic acids which are made up the genes. So these mutations created in a gene. Okay, so I'll see ATCG. So this can change to ATT. C can be muted into T. Okay, so so those kind of change can be done. So so this is when this mutation is done on this particular nucleus, and if this cell decides to divide, okay, so what would happen? Cell now with muted genes decides to divide. So it creates two daughter cells with this muted genes. So this is done via a pathway. Okay, so in a pathway we have certain proteins that would signal out different genes to express different genes. Okay, so as an example, when the UV radiation hits and change is done, however the cell doesn't know about this. So then the nutrient signals come. So nutrient signals come, and the proteins which would take these nutrient signals would carry out the information to the nucleus. Okay, so this is a protein 1 which acts as a messenger protein, takes this information that nutrients are there, we can divide. Okay, so that information is taken by this protein 1 to the nucleus. So when it's taken to the nucleus, so in the nucleus there are certain other messenger proteins which would take this protein and activate the these division proteins, division related proteins. Okay, so once it becomes the division, so there are certain other proteins as well that would incorporate the division process, help the division process. So this is protein 2, and there's protein 3, and in this pathway there's a protein called nucleic acid division related decision making proteins. So, so those would also be in this pathway. So a pathway is a set of chemical components, maybe proteins, maybe enzymes that would help out this process. Okay. So you can see in this pathway, cell division pathway. you have certain proteins here, which would ultimately ask the cell to divide or not. Okay, so that division is also actually checked by the guardian human gene. There's a protein called human genome, the guardian of the human genome. Okay, so that protein, so which is actually P53, one of the guardian proteins. If you are interested, you can read about this. So this protein is also one of the component of this pathway. So that genome checks whether all the genes are correct, okay, whether there are any mutations done. So that is actually being checked by this guardian of the human genome protein. So this is one of the guardians. There's another two or three. So this checks that whether they are actually in order, and if there are any abnormalities, if it would not allow the cell to divide, it would not allow the cell to take the decision for division. Okay. So there are mechanisms like that. Also, I told you that information because maybe I thought you maybe it may be interesting to hear. So, so there are things like that also. However, the main basic objective of this actually to introduce vata pathways. So in this pathway, you can see in the cell division pathway, there are certain proteins and enzymes involved in the whole process, okay, one after the other. So that's a pathway. So there are different pathways like cell division pathway and human whatever this every cycle is related with pathways, and for every purification process done, every manufacturing of these components and genes, all are related with pathways. So if you look at these databases, you can see those pathways. So from these pathway databases, KEGG is very popular. You can have a look at that pathway. Okay. Look at pathways in this KEGG. And there are annotation databases. So what is annotation? Again, I will share the screen for the whiteboard, and so what annotation is actually, once you have a sequence, ATT, GGCC, something like this, okay. Once you have a sequence like this, annotation is actually identifying important locations or features of this particular nucleic acid sequence. As an example, we can say we identified that from here onwards, this gene starts. Okay, from this place onwards, gene starts, and this is the promoter region. Okay, so that's one example region. So from here onwards, this is the gene, and this is the promoter region where the other compounds can bind to this region, and after that, after those compounds bind, only the expression starts from here onwards. Okay, so this in this region is the region where these other compounds would come and attach. So this identifying these the place, okay, and identifying the promoter region, how many characters are there, how many nucleic acids are there, and how long is the gene, from where does the gene start, and where does it end, and what's the ending sequence. So is there any ending sequence? So those things are actually identified in annotation process. Okay, so those annotation related data are stored in these uh, annotation databases. And now I think you have some idea about all of these databases. So if you have any problem, you can have a look at the web or ask me. So I'll try to help you. So actually in the coming practical session, so there are some related exercise for you to understand more about these particular databases. So however, I don't know whether we'll be able to do it next week. So this is about all the different types of databases. So we are going to now talk about sequence databases, which is one type of database available. So the sequence databases actually again can be divided like this, or sequence databases which contain all type of sequences. Okay, all type of primary sequences. Once a researcher, experimental biologist find a sequence, he would upload that sequence into GenBank or EMBL Bank or DDBJ. So which of these three are hosted by these three organizations in three countries in Japan? So here this one in UK and USA. Okay, so NCBI has all sequence database GenBank and clean non-redundant sequences are reference sequence. So why do they this clean and find out non-redundant sequences? Mainly because in GenBank many experimental scientists, any experimental scientists, even me, even you can upload any sequence. Okay, so the clean and non-redundant sequences are filtered from this GenBank and uploaded into reference different database, which is called reference sequence. So it's available in NCBI. Okay, NCBI's clean non-redundant sequences are maintained in reference sequence database. Okay. So an mRNA related database are again taken from GenBank and kept in Unichi. It's done by NCBI. Again, protein-related data of any protein sequence you want to know, if it's available, it can be downloaded from protein, which is again hosted by NCBI. And this EMBL bank, which is hosted by UK EBI, they have protein-related data stored in their UniProt database. Okay, DDBJ is the primary sequence database hosted by Japan. So if you look at this, I know you have some confusions because anyone can upload to any of these three databases. Okay, how these databases work together. So that's actually an interesting question. So actually, these all three databases share their data. So at the end of the day, all the databases contain the same data. Okay, in USA, it uploads data to GenBank, and in Japan, uh, not only Japanese people do it, all people around the world would upload through DDBJ. And in Europe, people can upload using this European database. So they would differ only in the organization of the data and the tools and database links available. All three would contain same data because they are updated. So looking at the NCBI, it's established in USA in 1982, and it's a national resource funded by the USA government, and it has a series of databases, and it would develop software tools analyzing for analyzing genome data. If you log into this database, you can see that it would disseminate biological information. It conducts research in computational biology. So there are researchers attached. So this is the NCBI website. So I want you also to log in and have a look at the front page of this website. So this is the NCBI website. So if you click on, I think you can see, can you? Okay, so you can log into this website using NCBI, NHM, NLM, NIH, GOV website. So you can just Google search, type in just NCBI, and there you would go to this page, and in this box you can see all the databases available. So we talk about gene, isn't it? So gene is a database where all the genes are available. And so genome is another database which would help us to find out about genomes. So let's find COVID-19 here. COVID-19. So I'm finding in genome database COVID-19. Yeah, it went to severe acute respiratory syndrome, coronavirus 2. Okay, so let's see, this is a summary. It has the length is given, protein count is given, and you can see there are 12 proteins and 12 genes available. And if you click on this reference sequence link, it would give us more information about the definition, the name of the virus, Wuhan H1, the complete genome is available, and the organism. So you can see here the organism. So starting from viruses. So this is actually the tree, and this is taken from the tree of life. Okay. So as I mentioned you earlier, this tree of life has several branches. One is eukaryotes, one is bacteria, one is viruses. So this Wuhan virus, it is coming from virus origin. So that's the origin. And from there it deviates into these viruses, and so at the end it comes to this virus. Okay. So after that, coronavirus appears. So it's something like this. So once you have this tree, if this is the tree of life, so we are in the eukaryotes. Okay. So this is the virus branch. So sorry. Hello, lecture lecture. Oh, lecture online. Okay, so so there are different branches available. So we are in eukaryotes branch, and this branch is for the viruses. So we start from the virus, and you know the tree of life. It has several different branches like this. So if the coronavirus, now we can see here that that listing shows you viruses, and then A, and then B, and then coronavirus here. Okay, it will say V, A, B, C. So that sequence of generations is shown here. Yes. Can you see?
Yes, madam.
Yes, madam.
Okay, so you can see here that A, B, C, D refers to here virus, riboviria, likewise. Okay, so that tree, the corresponding grandfathers, fathers, and the parent links, all of those links are sequentially written here. Okay, so that's the relatedness with the top organisms. So So there are different these actually data is based on references. It means scientific publications. So the title is A New Coronavirus Human Reproductive Disease in China. So which is published in Nature, which is a very prestigious journal. In 2020, it was published. So from those several references they have followed, and they have got the data, and finally it would show you the at the end you can see the genome, whole genome of the coronavirus from here. Okay, so you can download this also. So when you are doing the practical, this will be actually familiar to you. So when you are downloading, what you have to do is you have to click this, whatever the format you need. I clicked on FASTA format. That is one of the formats of this genome data. So then you can download the complete record as a file to your, you can download, okay, like this. So that's some other information. You will be actually familiar with this when you are doing the practical session. So I just showed you that all the available genomes or whatever the protein sequences and interesting gene sequences can be viewed and downloaded like this. Okay, let's look at a gene, maybe P53, which I mentioned you previously about the guardian of the genome. Yeah, P53 from tumor protein P53, which is available from Homo sapiens. So I checked it in gene, okay? So I'm looking at NCBI gene and P53. So this is in Drosophila. So that there also you go to the particular information page and you can have a look at the information, okay? So let's look at that later, and let's now move to our slideshow again. So now you can see this NCBI website page, isn't it?
Yes, madam.
Okay. So we talked about nucleic acid, so NCBI. So there we talked about this all databases link where you can have a look at many information databases available, information about databases available. So we are now talking about some information about these databases. So one of the databases is GenBank. So in the GenBank sequence database is again open access. It has an annotated collection of all public level nucleotide sequences. And also they have their protein translations as well. So, so these are directly submitted by individual laboratories, okay? Maybe bulk submissions also from large scale library, laboratories, you know. There are some university related laboratories which would include information about many genes. Okay, so they are uploaded frequently. So GenBank it continues to grow at an exponential rate, doubling in every 18 months. So so that this information is actually available here as well. So if you look at the web and if you find out information about this growth, you can see here how this GenBank basis grows. In 2020 also there's a huge increment, and from high 1985 the number of sequences also grows, and it mentions that it doubles approximately every 18 months. Okay, so this is information today. So So, so these, so about the annotations, the record is divided into sections, subsections, okay? So this is about one of the information about mRNA, PCCX1, Homo sapiens mRNA for protein contains CXS1 domain 1 complete coding sequence, CDS mean coding sequence. So which means in the nucleic acid there are certain regions which would code the proteins and certain regions which should not code the proteins. Okay. So this CDS mean complete coding region is available in this record. So this, I think I discussed about some of the sections of this data record. So this is a gen bank record. So you can see the source organisms which we talked. So this is about Homo sapien mRNA. So we start from eukaryote, you can see. So previously about COVID-19, it started from viruses, okay? So it starts from eukaryote and it ends at Homo. Homo is our type of organism name, okay? And there's accession number, version number, these are the numbers which would uniquely identify this particular record, okay? Definition is given. And there are references available in each and every, for each and every reference they referred to take this information, take the information they required, they mention it here. And the location of the regulatory region, coding sequence, exons and introns. So those sequence annotations or features are available here. Okay, gene, where is the gene? From 1 to 2486, gene is available from 222. Sorry, coding region is available from 222 to 2199. So this is region. So if you click on these links, it would highlight the particular region of the coding region at the end. So you know, at the, if you scroll here, I told you at the end you can find the whole mRNA or gene or whatever interest record explains. Okay, so here the record explains about the mRNA. So the whole mRNA is available at the end of this record. Okay, so as an example, if we go here, the whole record is available at the end. You can't see this, can you? So that's why sometimes I talk and you can see the screen. So that's why I want someone to talk with me frequently. Okay, so now you can see this, isn't it?
Yes, madam.
So here you can see at the end of the record, you can see the whole genome of COVID-19. So we know the coding, we want to look at the coding region only. When I click on that CDS, it would highlight the particular coding region. So this is the only coding region for proteins or whatever the interested proteins. Okay. So the translation is also given here. It will translate and create proteins from this particular region. Okay. So that's why actually I mentioned here. So it's quite difficult to do the lectures in this way, you know. So okay. So so the cross references to links to the records of other databases are also given here for the interested one to have a look at the whole information available in the web. Okay. So so these are the common sequence annotations. These are the common sequence annotations as I previously also mentioned. So if this is the genome sequence or a gene, it would identify where the promoters are and where the terminator of that gene and which part of that is the coding region. So this is the coding region starting from here to here. And the gene sequence, so you know from the whole gene, all these coding region would not create protein. Some of those regions, the green regions, do not code proteins. It's like that. Okay, so it's that's there for a purpose. to create the vast heterogeneity in the proteins, and also to create a large amount of proteins from a single coding region. There are some regions included in middle which do not involve in coding. So these exons, which are shown in blue color, would only create the protein required. So this is the protein, and this is the translation process you discussed last week. So these annotations is something else. Annotation rather. Annotation is the process of identifying these promoters, terminators, CDAs, exons, introns. So that is called annotation. Okay, identifying the important regions of a gene. So those annotations are available in these records for anyone to understand. And there are different IDs. If you see in this record, accession number, version number. So what are they? And there's a gen bank. Again, there's a number here. What are they? So that's what we are going to look at now. So GI, gene information identifier number, is the GI number. So there's a GI number here. So it's the gen bank identification number. So the GI number is a series of digits consecutively assigned to each sequence record process. There's no logical representation or implied meaning, just a serial number. Whatever the uploaded sequence is, that's an auto-generated serial number. Okay. However, it's unique for each and every record. So if you assume that you are an experimentalist, you uploaded a sequence, and once few days you found out that there's a problem in this particular record, okay, and you corrected that, and you have to upload it again. When you're uploading it again, this GA number would uniquely identify that. Okay, it would not relate that to the previous number. It would give you the particular number. Thus, this is the unique identifier. Like that. So this is actually used independently by GenBank and ENA to uniquely identify the records. And the accession numbers are something different. So accession numbers would identify the sequence uniquely. Okay. So if there are two versions of the same sequence, they would have the same accession number. It's not like the previous GI number. So as I told you previously, if someone uploaded some sequence and correction at the second time, this accession number would have two versions, but the same number. Okay. So it is consistent across all databases. So these three databases, which are hosted by USA, UK, and Japan, they would have the same number. Okay, they do not have different numbers for the same version of the sequence. And the version number is something else. So it would represent and uniquely identify each correction of a sequence. So if there's a corrected version, it would have number 0.1, something like that. Okay, so this is the accession number. After that, there's 0.1 if that's the first version. If that's the second version, 0.2, like that. It would represent and identify each correction of a sequence. It would be incremented every time when the sequence is changed. So if change is made to the sequence, it would receive a new GI number, and the version number would be increased. So let's look at this, whether there are any version numbers available. I think you can see. Okay. I'll share the screen. Okay, so here you can see those numbers. Okay, in CPI reference sequence number here, and version and version number, sorry, accession number and version number. So this is the second version. You can see it would say 0.2. So that is the version number available. So so that would say that this is corrected twice. Okay, so this is the second time it's available. And so that's about the numbering. So you know now there are GI numbers, accession numbers, and version numbers available. And reference sequence is another clean, high quality version of GenBank. So we up to now discussed about the GenBank, which should store primary data. Okay. So now we are going to discuss about secondary level data bases. So before starting that, give me a few seconds break. Actually, I want to. My laptop is running battery low. I want to turn the battery on. Okay. Give me a few seconds. You can take a break of maybe 2 seconds, 2 minutes. Sorry. Okay, can you hear me? Can you hear me? Hello?
Yes, madam.
Okay. So is everybody is here? The others?
Yes, sir.
Okay.
Excuse me, madam.
Yes.
I have a question, madam. In
Okay.
previous slide, the madam taught us about the accession numbers.
Yes.
So there can be two entries with same accession number.
No, the version should be different. If that's an update,
Okay.
it should be different. The version, the accession number, so after that there's a point something and it means there are two versions of the same accession number. So same gene, there are two versions. So like software, you know,
Okay.
software, two versions, slight differences. If that's the same gene, the accession number is same. Is that okay?
So, but madam, I don't understand what is the relation between the GI number and accession number.
GI number is something like this. So if you're an experimentalist, okay, you just upload a gene, gene A, okay. So I uploaded gene A and so after some days I found that there are some errors in that gene, okay.
Okay.
Identification process, there are some errors and I have identified a more correct version and I upload that, okay.
Okay.
So now gene A they have given a number to 2020, okay.
Okay.
When I upload in the primary sequence, the same sequence, they have given 2020 and the second time they give me 2051 something, okay. Just
Okay.
serial number they generate.
Okay.
Okay. However, the same gene is there. Okay. So after the identification of the gene, let's say that is gene A. So they give an accession number for that gene. Okay. Let's
Okay.
assume that the given accession number is 2040.
Okay.
So after that in 2040, so that's the accession number, real genes, real identifier is that. However, for that gene, there are two versions available. Okay, version 1, which is accession point 1, version 2, accession point 2. Okay.
Okay, madam.
Okay.
Thank you.
All right. So let's now move on. So this is actually about secondary level data, reference sequence, which includes clean, high quality version of GenBank. So in GenBank, there can be repetitions. Okay. Maybe the same gene would be uploaded by two people. However, there are two GI numbers given and it's identified as two records. So in this reference sequence database, RefSeq, it only contains this high quality clean data. Okay, it is a in short term, it's a clean high quality version of GenBank. It's a collection of selected nucleotide and protein sequences. It contains sequences and annotations. It means it has, if there are regions identified, that information is also included there. And there's no redundancy, that's the important thing. So it would contain only one sequence for each genomic region. And the curation is done by people at NCBI. Okay, they would create most complete accurate data from GenBank. It's a very difficult thing. However, they do it, and this database capacity increases. Okay, so capacity actually, the amount of data available in this database increases day by day. So it means these people at NCBI are actively doing that. So it's free to use and no redundancy. So this is one of the important and useful databases for us. So this is a clean high quality version of GenBank. So if you go to NCBI and click on this reference sequence, you can select that. So you can have a look at that later. And so next, we are going to look at the differences between GenBank and the reference sequence, as I mentioned previously also. So this is GenBank and this is reference sequence. So if you look at the similarities and differences, so the first one is primary sequence database, and second one is clean derived data included database. So in primary sequence database, only one submitter can make corrections. Who is the person? Who is actually the person who uploaded the sequence? Okay, there's only one submitter, and no filtering. All the sequences available there. Redundancy is there. Okay, two people may have uploaded the same sequence. Who knows? Okay. However, the updating to the sequence can only be done by the person who uploaded. And in reference sequence, provider can derive or correct data, and filtering is done on organisms, and it has a very good quality. And there's no redundancy available. So if you look at this reference sequence, once you compare this with primary sequence, the major important difference is the redundancy. There's no redundancy available in the reference sequence database. So now let's look at how to retrieve data from NCBI. So I have already shown you that using point 90. So you can go to the home page, and there are dedicated pages as well. And if you want to do a cross database search, there's a way called jQuery. So you can go to the jQuery and type whatever the word or name of the protein or name of the genome, or if you don't know what that particular name is, also you can search. There's a cross database search. So it would search on each and every database and it will give you some corrective result. So you can actually do this when you are doing the practical session. However, if you're keen and if you want to do that, you can do it by your own. So in CBI website, how to search nucleotides. So actually here you can go to the nucleotide. Let's do one. I think we did for P53, isn't it? So here you can see, I think you can't see it. I'll share the screen.
We can see, madam.
You can see my website also.
Yeah, the website.
Okay. You can choose nucleotide and then type whatever the interest in gene. I'll type P53 here because we searched that earlier as well. So it's available in different records. There are different records available with that particular gene name. So here you can see this filters available in both sides. You can see top organisms. In these organisms, this amount of records available in this particular page. So if I'm interested in Homo sapiens, I can click on this. So then that will show you all the records from Homo sapiens. Now you can see Homo sapien mRNA. And molecule types you can search if you want mRNA, cDNA. So likewise there are molecular types interested. You can click on tick on them, so that will show you the corresponding results. And source databases, you can see in reference sequence because I would be interested in reference sequence because there are no redundancies. So likewise, you can filter out whatever the data you require. Okay. So after that, you can go to the record you like. And again, you can see these accession numbers, version numbers. This is the 11th version. It has gone through many revisions. And at the end, you can see whatever the, what is this? We are looking at a chromosome, isn't it? Yeah, so whatever the chromosome or whatever the gene data. So, so there are protein and gene sequence formats. So that's how we can search. We have done two searches about P53 and the COVID-19 genome. So likewise, you can also try to find out a protein. So I think later in this slideshow, there's exercise to find a protein. So let's do that later. So actually to have a look at these sequences, one should know what formats these sequences are written. There are several formats available. So from them, so these are the formats available. GenBank, EMBL, they have their own formats. SwissProt, it's again a database which contain information about proteins. It also have their own format. FASTA is the standard format. and genetics computer group, they also have one. Okay, FASTA is standard format, so let's talk about that. So in a FASTA sequence format, you have in FASTA sequence format, you have the first line start with this greater than sign. Okay, then it's followed by a sequence identification code, and there are one or more lines containing sequence. It's something like this. So let me explain it with the whiteboard. So it starts with the greater than sign and some information about the nucleic acid is given here. Then after that the nucleic acid starts until then it can be repeated. Okay, not repeated, it can be written. So so how can we identify how many nucleic sequences are there? So this is the first sequence, and if there are several sequences in a file, can you propose a method to identify how many sequences are there? Hmm?
Add a new line after the sequence.
No, actually. So you know every sequence start with the greater than signs. You can count simply the how many greater than signs are there. Uh, so that's very simple. Okay. So a file in FASTA format may comprise one or more sequences. So so that's why we have this sign to identify each FASTA format, FASTA sequence. So it's called Pearson format as well. So these are the nucleic acid codes which are available in nucleic acid sequences. So I think we discussed about them. So these are the amino acid codes available. And this is a simple FASTA sequence. So you can see here, the GI number O is given here and some information is given here. So this line soon after the greater than sign is not processed. So the sequence starts from the next line. Okay? So you can count the number of FASTA sequences available by counting the greater than signs available. So this is another format, GenBank FAT file format. So you can see it starts from locus, the sign letter locus, and the definition is letter origin, not locus. Okay. Origin. So after the origin, it would give you the nucleic acid sequence. So it starts from the locus, which is the number, and the start of the sequence is marked by this line candidate origin, and the end is marked by double slash. So that's actually a P53 suppressor. We already found that. And GenBank FAT platform, you have already seen this. In COVID-19, they have given that genome sequence. I think you may remember starting from origin and ending with these double slashes. So there are search queries you can see. Once you find, once you click on these filters, and it will create a query like this, because you know this is a database searching. So here it will show you the query. This is all fields, find this one, and Homo sapiens as the organism, or Homo sapiens in all fields. So likewise, the query is given here. You can have a look at that. So when you're finding, okay, when you click on the filters, like as I did at the in that At one side, you can see this query here. So to understand the search queries, you can also type the query. So the organism means the preceding search term Homo sapiens is searched only in the organism field. So this is the organism field. Okay, you may just remember this. The organism, according to tree of life, the organism's history is given here. So, so this Homo sapiens word would be searching organisms. So since you have given this here, the word only would be searched from the organism field. And it would only return human records with the organism equals Homo sapiens. And there are Boolean operators used, and is used to narrow down the search to retrieve records contained in each term, and not to retrieve records that are not in the predicate term, or to retrieve records contained in any of the terms. Okay. And these Boolean operators are always proceeded from left to right, So here, if you use O and not an O like this, it would be, I understood by the database search like this. The records that contain either promoters or response elements from this list, get that list, and from this list remove the records that contain human, not human, and then add records that contain mouse likewise, from left to right. Okay. And you can use parentheses. So you always parentheses have the high priority. So when you are searching, you can use these logical words or parentheses. And wildcards also can be given like these stars, all, okay, all fields. To represent all fields, you can use stars. So if you're still getting many records, you can use this advanced search option or filtering out. We have done filtering out. Advanced search also, you can look at. It's easy to find out. You can see here. You can just click on that and do advanced search if you want. So once you are doing advanced search, actually you should have very clear information about what you are going to find. So finding all genomes, we found out COVID-19. We can have a look at HIV-1 also if you are interested. I'm just moving on because we have some more to discuss. So NCBI also have a cross-query search, cross database search. So here, in NCBI, there are several databases available. So as I showed you this in the first page. So this gives you all databases link. Okay, so you can go to all databases and find COVID-19. Then it will show you all the databases results about this COVID-19. Okay. So it is available in these databases, literature databases, gene databases, genome databases, and clinical databases. So this shows you the amount of records available in those databases. In genome, there's only one. Okay? There's only one genome database entry about COVID-19. So likewise. So these are some of the databases available in NCBI. So these are not the whole lot of database available. You can see here there's a very large amount of databases available. So I can't list them in a simple one slide. So here are some important databases taken into this. So some of them are actually this midline, which is a literature database. PubMed, again, a literature database, important literature database, and databases of human genome. So what is a literature database? So I think that would be important for you since some of you are doing bioinformatics projects. So if you go to NCBI, this PubChem compound PubMed is actually the database which contains literature. Literature means published papers. So if you write my name, so you can see the publications related. This is author name search. Okay. So likewise, whatever the information. So let's see one. So you can see my publications here. So likewise, if you write about, if you want to know about COVID-19, it is very interesting topic these days. So, so you can see that whatever the articles available. So this would show you the full text available, whether it's a PDF availability or not. As and if you click on one, you can see Elsevier full text article available, PMC full text freely available. So like us, it's very interesting and important database. Okay. So that's about PubMed. So Medline is also a literature database. OMIM is a database of human genes and genetic disorders. So if you want to look at any information about genetic disorders, this contains mainly the nucleic acid sequences related with those genetic disorders. Okay. So OMIM is one of the important, very important database available by made available by NCBI. GenBank, we have talked about this. It contains all publicly available DNA sequences. And protein database is a database of amino acid sequences from these databases and translations from annotated code ingredients in GenBank and reference sequence. So you know in GenBank it contains genes. Okay. So likewise, the protein database contains information about proteins translated by the genes in the GenBank. Okay. So genome database contains information about genomes. PopSet is a database of DNA sequences that have been collected to analyze evolutionary readiness, evolutionary relationships. And taxonomy is a database of names of organisms. You know the different names. They use different names. They use the biologists use. And PubMed, we looked at the PubMed and we did several search. The US National Library of Medicine's Digital Archive of Life Science Journal Literature. It contains literature of life sciences, okay, related data. So the access to full text articles in PMC is free. So if journal requires a subscription of some money to access data, it will not be available. If it's not, all the articles will be freely available from PMC. OMIM is online Mendelian inheritance in man. So it's a database that collects all known diseases with genetic component. And it's actually began by this doctor at Johns Hopkins University. It's a good place to start with research which contains disease or a biological molecule which has inheritance related information like inherited disease. Okay. So how to submit a sequence data to GenBank? Actually, there are two main methods: banking, which is a web interface, and a sequencing, which is a program. So why do you think they have web interface as a program? So why do you as software engineers allow these two? I mean, you have web-based thing and you have a application program that need to be downloaded and installed. Why do you have these two versions? Hmm? You are now partly a software engineer, isn't it? Can you hear me?
Yes, madam. Can you repeat that? I didn't understand what.
So for certain programs, there are web-based program, and sometimes we have to download the program and install and use it.
Yeah.
Do you have two versions like that?
There might be some system requirement and some, some might have a low powered laptop or something.
Any other?
Hello.
Yes, any other? No, that's not the correct answer. The main reason here is actually so when I mean the things we can use, things we can do in a web-based program, web-based interface is limited. Okay, so here in Sequin, if your DNA sequence is very long and bulky, okay, it cannot be uploaded by banking. It's a web-based interface for limited length sequences. But Sequin program, you can use it very large site data sets to do bulky uploads with maybe several copies of same genes or several genes. In bulk form, you can do uploads. So that's the main difference. So I see that also same in the other web-based interfaces. The things we can do is limited, isn't it? However, when you download it and install it, it can use the full capacity of the whatever the machine, and you can do many things than you do in the web interface. Okay, that's the main difference here in this application. So finding proteins. So there's a exercise for you to find the human prion protein. So may I ask you that as a homework? You can find the human prion protein and try to download it, and maybe you can paste the sequence in Notepad and upload as an assignment. Okay. So I'll put the LMS link for that, and I think this video, I don't know whether I will be able to upload this. Okay, if anyone is interested, I can put to the Google Drive and share it with you. But I don't know whether the size would be enough to be uploaded to LMS. Now the LMS usage would be increasing day by day because of this current situation. Okay.
Yes, ma'am.
So I'll put the LMS link, and next week I don't know whether I'll be able to do the practical session by next week. So let's move on to the next topic. Okay. And do the practical later. Yeah, that should be done in that way because it's quite difficult to do it online. Because I don't know what you do, At the at the university, I can come near you and help you and do the lesson. And in that practical, there are some group activities also. So let's postpone that practical session. Whenever we start, we can do that. I think now you have some idea to retrieve information from these databases, isn't it? So
Yes, ma'am.
you can collectively, I mean, if there are any problems, you can discuss with your friends. So I should tell you that this fourth year is, I mean, you should get together and discuss things. I mean, when we are doing the fourth year, we were always together. Okay, at the university, if that's a Saturday or Sunday, uh, we were there. We were there discussing things, and We had fun time at the gym also. I mean, we were together. I mean, this situation, you can't do that. However, you can do it online. Maybe 1 hour or two, where you can discuss problems you have while you're absorbing the lessons, and you can discuss things, okay? Make the things clear by, I mean, from the, after you discuss, after the weekly or daily, you make the things clear up with your friends, and I mean, there's no any challenge at the end if you do it. Okay, so that's up to you. I'll stop from here, and I'll upload the link to, I'll put the link in the LMS. I think all of you, many people have emailed me the previous whatever the program you wrote, but I didn't have time to check it. I'll check it and let you have my feedback. However, if you get the corresponding, for the corresponding, if you enter the corresponding nucleic sequence, and if your program can convert that into a particular protein sequence, that's it. Okay. All right. Thank you very much, and have a good day.
Thank you, madam.